Hi everyone. My name is Ben Klein. I'm a graduate research assistant at Michigan State University. I apologize, I'm getting over a nasty cold, so you'll have to bear with my voice for this presentation. But thanks for having me. I'm excited to share some of the work that we've been doing. Um, just a little bit of history behind this work. So back in 2018, there was a charge from the Lake Superior Committee to the LSTC to assess the potential genomic and phenotypic impacts of Splake on the recovery of native brook trout and lake trout within the basin. And so, in collaboration with a bunch of partners around the basin, we've developed a project to assess this question. There's three primary aims to the overarching project, but I'm only going to focus on aim 2 today, which is to characterize the potential fitness impacts of splake introgression in known crosses. And so there are three primary objectives to this work. The first of which was to create experimental backcrosses between brook trout, lake trout, and splake in a controlled environment. A big question is whether or not splake are sterile and if they can actually reproduce back with brook trout and lake trout in the wild. And so we set up an experiment to do that. We wanted to assess if these crosses were viable, and then if they were, examine what the genetic effects might be. And just to start with a really high-level background, I'm going to talk a lot about hybridization, and you might hear me say introgression, and I'll use those terms interchangeably throughout, but really what this refers to is just the interbreeding between genetically distinguishable groups. And we care a lot about hybridization and introgression in biology because it has a really influential capacity to shape the genomes of organisms. And actually, over 10% of the extant species on Earth are the product of these ancient hybridization events. And so, in a management context, we're really interested in how introgression shapes genomes and affects organisms and their potential fitness down the line. And so, as many of you know, splake are a cross between brook trout and lake trout. It's important to note that they're what we refer to as an anthropogenic hybrid, which means that they're not naturally occurring out in nature. They're produced in a hatchery and then stocked out. In Lake Superior, they've been stocked over the last 50 years, and this was initially to provide an important recreational fishing opportunity amidst declining lake trout populations. However, despite the long history of stocking in the region, we have a limited knowledge about the potential effects of splake on wild populations of brook trout and lake trout. And so that brings us to the experiment. So we wanted to create these controlled crosses between brook trout and splake, lake trout and splake in a hatchery environment. We crossed both parent species, and then we also had control crosses between brook trout, and lake trout, so we could use them as a reference. We included the male and female reciprocal crosses so that we could account for the effects of sex in our crosses. And overall, we had 3 replicate within each cross for a total of 18 experimental families. And so essentially, we did our crossing with the gametes that we had collected. We reared those fish up to an early juvenile stage and then terminated the experiment at 90 days post-swim up. Then we collected tissue samples from all of the surviving offspring, and we used restriction site-associated DNA sequencing, also known as RADseq, to survey the genome-wide genetic diversity to look for signs of introgression in the offspring. And so here's a closer look at what our experimental families looked like overall. So as you can see, we have our 3 replicates within each cross. Notably, we were unable to find a third reproductively mature female splake. So in our male parental species and female splake crosses, we just have 2 replicate families instead of 3. I wanted to just offer a really sincere thanks to the folks at the Les Voigt State Fish Hatchery. We conducted the experiment there, and they did a tremendous amount of work to get this going, including caring for the fish, enumerating the offspring, and all of that. And so this wouldn't have been possible without their help. So as I mentioned, we collected tissue samples from all of the offspring, and then we conducted genetic sequencing. Really, all this boils down to is looking for what we refer to as a single nucleotide polymorphism, or a SNP. And so these are just these single base pair differences across the whole genetic code of an organism. And on this example on the slide, there are, let's say, 2 fish from a blue population. Maybe those are the brook trout. And they all have an A at that specific region of the genome. And then a green population, let's say those are lake trout, they have a T. And so really all we're doing is surveying thousands and thousands of these regions across the whole genome to be able to answer our questions of interest. And so our final data set here was comprised of over 480 individuals. That includes all of our replicates and our 16 families. We genotyped all of those fish at around 11,000 regions, like I just described. I'm pleased to report that in our final data set, we retained over 25 offspring for each family in our crosses, which was great. And then we also importantly had all of the parent genotypes, which we could use for a reference. And so getting into our findings, first and perhaps most interestingly, we did find that Splake do produce viable offspring when you backcross them with brook trout and lake trout, at least in this controlled hatchery environment. And so here's some photos from our experiment. And as you can see, we have some fully formed fish, which I think is great evidence in and of itself. And so now that we found that the crosses were viable, we wanted to examine the genetic variation underlying each of the offspring. And so the first thing that we did was just take a look at the overall genetic variation in all of our samples. I know there's a lot going on in this plot, but I'll show you everything that you need to know. In this analysis, each point is an individual fish, and the closer the points are together, the more genetically similar they are. And here the color coding corresponds to the cross design, so each color is one of our experimental families. And so the first key takeaway here is that our brook trout, lake trout, and Splake are all genetically distinct. So brook trout over there on the left-hand side of the screen, you can see they all cluster nicely. Same with the lake trout on the far right, and then Splake actually fall right in the middle, which is what we might expect since they're half brook trout, half lake trout. Things get a little bit more interesting when we look at their offspring. And so our hybrid cross offspring, our back crosses, actually show a lot greater genetic variation than any of the parent species individually. And so over on the left-hand side of your screen, those are our brook trout Splake back crosses. So as we might expect, they fall somewhere in between brook trout and Splake, and then we see the mirror in our lake trout as well. And so we thought this is interesting. There's something going on with the genetic variation. And so we wanted to dig into this a little bit more deeply. So the next thing that we did was conduct an ancestry analysis, and this is essentially a way of looking at the proportion of ancestry to brook trout and lake trout. So for example, here in this plot, each bar is an individual fish, and then the y-axis is the percent ancestry from 0 to 100. So if your bar is all one color, for example, blue, that fish would assign 100% brook trout. Same for lake trout. If you're 100% orange, you're 100% lake trout. And so our controls look great. Those are just known brook trout crosses and known lake trout crosses. They assign excellently to their parent species as expected. Interestingly, if you look on the far right, those couple individuals at the end are the Splake parents in our study, and they have a 50-50 brook trout, lake trout ancestry, which is what we might expect. And so we would expect then that their offspring, if you crossed 50-50 brook trout, lake trout with 100% brook trout, you'd have something around 75-25. And so that's what we would expect based on normal inheritance patterns. And actually what we find here is that if you look around that 75-25 mark in our back crosses, it actually doesn't match our expectation. I'm going to make this a little bit easier to see on the next slide. So I pulled those ancestry coefficients from the analysis that we did on the previous slide, and I just plotted them in a frequency. And here you can see that The dotted line is 75%, which is our expected ancestry. And in both our lake trout backcrosses and our brook trout backcrosses, we see that the mean is actually favoring the parental species. And so what this tells us is that not that there necessarily aren't any fish that are 75-25. You can see some that are kind of shifted to the left, but this might be evidence of what we refer to as viability selection. And that means that when those gametes were formed and brought together, all those individuals that were in this expected ratio might have actually died. And so important to note that everything that we sampled at this point in time, at the 90 days in our experiment, was what was still alive. So we know that the ancestry is shifted, and we know that there's differences in the genetic variation in our offspring. And so we wanted to look at the transmission of genetic information from parents to offspring because we have this information in our crosses. And so we did this to be able to identify whether or not genes are being inherited in the proportions that we would expect based on what we know the parent genotypes are. And so here on this slide, I'm just going to show a couple plots that all kind of fall into the same general category. Really, all you're looking for here is that if a gene is inherited differently from what we might expect based on the parents, it'll appear above the dotted orange line as significant. And so I'm showing the results now for our controls. So these were our known brook trout and known lake trout crosses. And as you might expect, we don't see a lot of differences in terms of what's inherited. There's 3 SNPs in brook trout, and this might be just due to noise alone. When you've got tens of thousands of SNPs, it's statistically likely that at least a couple will fall out. But no major patterns in our controls, which is what we want to see. And this phenomenon where the inheritance is different from what we expect is what we refer to as segregation distortion. And so no evidence in our controls, which is great, what we want to see. So next we examine this in our offspring between a female splake and a male brook trout or lake trout. And as you can see here, these crosses also look almost identical to the controls. We've tested more SNPs because there's more variation in the data, but as you can see, most of them do not differ significantly. Things get a little bit more interesting when you have a splake dad. So in our male splake crosses, we found that the offspring actually inherit genes in much different proportions from what we might expect based on Mendelian inheritance alone. And so in our brook trout samples, we see that there are almost 400 regions of the genome that are differentially inherited when there's a male splake used in the cross. And in our lake trout, we see almost 150 regions of the genome that are differentially inherited as well. So our key findings from this analysis is that genes are passed down in different proportions from what we might expect based on random inheritance alone. And this pattern is only apparent, at least strongly, in our male splake crosses. And so knowing this information, we wanted to dig in a little bit deeper and see where in the genome these affected regions are occurring. And so this plot is really similar to what I showed on the previous slide. Here I've just partitioned all of the regions of the genome into the 42 chromosomes in our reference genome. And so this first is just the brook trout and splake back crosses. And here, any of the colored points that are above the line are ones that are significantly inherited in a different ratio than what we might expect. It's important to note that our inherited regions are not random, so it's not occurring all across the entire genome. We can see that more chromosomes are actually more strongly affected than others. And importantly, these blue points are regions that were inherited differently across multiple families. So that was the importance of including our replicates. Can see that there's specific regions that are consistently affected, especially in brook trout on chromosome 6 and 19. We did the same thing for our lake trout back crosses, and again, we find a similar pattern. We note that the inherited regions are not random, and in lake trout, especially chromosome 20 seems to be highly affected by Splake introgression. And so we wanted to dig into those regions a little bit more deeply, and so we just looked at the gene annotations for all of the regions of the genome that were consistently affected across our different SNPs. I want to point out that this is like a cherry-picked selection here. These SNPs were near the genes, but there was no functional testing done to determine if they're actually playing a role. But just based on proximity alone, these genes might be candidates for future investigation. But in all three of these genes, they seem to be pretty important for development. We have examples from the literature in neuro and cytoskeleton development, as well as smooth muscle cell differentiation. And so in general, it seems like these genes could be important for morphological development, but additional studies would be needed to investigate that further. And so our key takeaway from all of this work is that Splake can successfully backcross with the parent species, and there are genetic consequences for their offspring. If Splake are hybridizing with brook trout and lake trout in the wild, we have a lot better idea of the likely genetic effects. Particularly, we know that stocked male Splake have the greatest potential to disrupt our native genetic diversity. We identified multiple regions of the genome impacted by introgression and also several genes that may be implicated in development that could be affected. So excited to report that this study provides a really valuable catalog of backcross samples for being able to detect hybridization in the wild brook trout and lake trout, which is the final stage of this project, which we're currently working on. And then I do think that this study provides an excellent setup for future studies to be done in the lab. Going into this, we didn't even know if our crosses would be viable. And so recreating a more elegant experiment where you can actually control the number of individuals in each family and do more controlled experimental testing would be a really cool next step to actually implicate these genes in survival and fitness outcomes. And so with that, I just wanted to thank our many partners for contributing to this work and all of the ongoing work with this introgression project. And if I have time, I will take any questions.